Okay, welcome back. Last time we talked about cell theory. This time we're going to get into some scales in biology, so relative sizes of things and converting between units. We'll look at some different types of microscopy, following on from our earlier discussion. We'll talk a little bit about biochemistry and the role of understanding biochemistry in cell biology, and we'll expand on that a lot during this semester. And then we'll look at one biochemical technique. So that's what we're going to do in this video. One of the things about cells is obviously they're small, and so the things inside the cells are even smaller. And so you have to start to kind of get an understanding and appreciation of the relative scales of things, so that you can better understand how rates of change occur and kind of the mechanics of cell biology. Size is also important in terms of thinking about tissue and then organ formation. Mainly we've got to think about tissues at this level. And the reason it's important to understand something about scale and size in cell biology is that a cell cannot be more than about 100 to 200 micrometers away from a blood supply in a tissue. And that's because of the limits of diffusibility of oxygen and carbon dioxide and many other nutrients, obviously. Let's kind of look at the sizes of these cells here. Now we measure cell sizes in micrometers or micrometers, if you like. On the left there, we've got a plant cell, average plant cell. They're kind of cuboidal. This one's a little elongated, 20 to 30 micrometers, and you can see the relative sizes of the chloroplasts. And animal cells, there's a lot of diversity in the size of these cells, whether we're talking about plant cells or animal cells. Massive diversity in the sizes of animal cells. From very, very, very small cells, 5 microns in diameter, all the way up to frog eggs, which are 1 to 2 millimeters in diameter, and you can see them easily with the naked eye. So huge diversity there. You can see an animal cell, 20, 25 micrometers in diameter. Obviously, it's hard to kind of discuss these sizes in some cases because cell shape varies. Now, if a cell is roughly spherical, then we can talk about its diameter. If it's relatively cuboidal, we can talk about its X, Y, Z dimensions. And so you've always got to think about the kind of size, the dimensions of the size that we're talking about. And then down at the bottom there, you've got a little bacterium. That's like an E. coli, maybe one micrometer wide and 2 micrometers in length. Obviously, much smaller there than a eukaryotic cell. So, but again, a lot of diversity there. You may have a Treponema pallidum bacterium. That's the bacteria that causes syphilis. It could be 10, 15 micrometers in length, but it's very, very, very slender, so very narrow. So maybe only half a micrometer, 1 micrometer in kind of width, if you like. So this micrometer that we're talking about, you can see it's indicated by the Greek letter mu, the lowercase Greek mu, followed by an m. That's a micrometer. Sometimes you'll hear me refer to this as a micron. That's a millionth of a meter. That's 10 to the minus 6 of a meter. So pretty small, and we'll get into this a little more in a moment. But that gives you some relative some ideas of the relative sizes of cells. So let's kind of think about this micrometer, the micron, and think about kind of its size relative to things you know. So a meter, so you can get an idea of a meter. If you're not kind of comfortable with the SI units, then that's something to work on. We're not going to talk about yards, inches, and feet. A meter is 1.1 yards, just to give you an idea, and a yard is 3 feet. But a meter is 100 centimeters. That's standard System International units. It's the SI unit system. 1 meter, 100 centimeters. 1 centimeter, 10 millimeters. If you find a ruler, it's easy to see what a centimeter is, and then see the 10 divisions there of millimeters. And so if you then have a millimeter, that is composed of 1,000 micrometers. So if we go from a meter to a centimeter, that's down an order of magnitude. So that's from 100 to 10, that's tenfold. And then if we go from 1 centimeter to 1 millimeter, that's also tenfold. Now when we go from millimeter to micrometer, though, that's 1,000-fold difference. So a meter is 10 times bigger than a centimeter, but a millimeter is 1,000 times bigger than a micrometer. So let's kind of put that in the context of some subcellular structures that you're familiar with. We've got some ribosomes, membranes, microtubules, microfilaments, also known as actin filaments, and the double helix there. So let's just kind of keep in mind down at the bottom here, let's see if I can get my pen going. Let's keep in mind that a millimeter is 1,000 micrometers, and in a moment we'll introduce the idea of a nanometer as well. But let's just start off kind of thinking about some of these structures that we know. Ribosomes, 25 to 30 nanometers. So a nanometer is 1,000th of a micrometer. So when we go from meter to centimeter, that's 10-fold. Centimeter to millimeter, that's 10-fold. Millimeter to micrometer, that's 1,000-fold. That means there's 1,000 micrometers in 1 millimeter. And then if we go down to nanometers, there's 1,000 nanometers in 1 micrometer. So that means there are 1,000,000 nanometers in 1 millimeter, because 1,000 times 1,000, that's 10 to the 6, so that's 1,000,000 nanometers in a millimeter. Now make sure you can convert in between all of these units. You know, I could ask you how many nanometers in a meter, how many millimeters in a micrometer, for example, that kind of thing. So let's look at our ribosomes over here. This gives an idea of scale. If we think about the large subunit and small subunit stuck together, total width 25 to 30 nanometers, that's tiny. That's in some cases very difficult to even visualize what that looks like. Put that in the context of a membrane. A membrane's anywhere in the region of 7 to 10 nanometers. It says 7 to 8 here, but you get variations in width based on membrane proteins. 7 to 8 nanometers is just a lipid bilayer, not necessarily a biological membrane, but a membrane, a bilayer kind of membrane. 7 to 8 nanometers. That's the width of the oily barrier which separates the outsides of cells from the insides of cells. So when you see these structures drawn in textbooks, and you've got this big, thick black line showing the plasma membrane, that's completely not drawn to scale. When you see these mitochondria and these dots showing these ribosomes and things, the scales are completely off. It's the artists doing their best to represent the size of things. Microtubules, microfilaments, obviously parts of the cytoskeleton. Now microtubules, diameter is about the same as the width of a ribosome, and you'll get about 3 widths of a membrane into a microtubule, and a microfilament there is about 7 nanometers. So start to get an idea of the relative scales of these things compared to the actual cell itself. So just think about a cell let's say it's a spherical cell, and let's say it's 20 micrometers in diameter. From that you could work out the volume, and if we then said, okay, look, the ribosome is roughly a sphere, it kind of isn't, but it's an approximation. We could work out the volume of the ribosome given a diameter of 25 nanometers. So we could work out roughly how many ribosomes would fit in the cell if the cell contained nothing but ribosomes. But if we then said, okay, 20% of the volume of the cell is ribosomes, then we can work out roughly how many ribosomes would be in a cell. So there are values there there is value, I guess, to understanding the relative sizes of these things. So make sure you can convert between these units. Let's keep those units up on the screen and then just take a quick look at some more relative scales. First thing to notice is that the size of object over on the left axis here is a log scale. So we're going from 100 millimeters to 10 millimeters to 1 millimeter. That's 0.1 millimeters, 0.01 millimeters, 0.001 millimeters. That's 0.1 micron, 0.01, 0.001, 0.00001 micrometers. So we're down, the rough radius of a hydrogen atom is about 0.1 nanometers. Now that's sometimes also called an angstrom. So you'll hear people talking about sizes of things in angstroms. 1 angstrom is 1/10 of a nanometer, or 0.1 nanometers. Let's go all the way to the top of the scale. Human height, well, average male, 1.7, 1.75 meters. Obviously a significant range there. You get the idea of chicken egg. But look, we've got a neuron which is longer. There are some nerve cells, some giant axons, which can be a meter long. Now they've got to be very, very thin. They're not a meter in radius. That's obviously silly, but they can be a meter long because they're very, very, very thin, so we can get a blood supply close to them. Frog eggs down here, 1 to 2 millimeters. Talked about those just now. Very large individual cells. Eukaryotic cells, getting a bit smaller down here. And And as we look at these sizes, it's important to bear in mind the ranges that we can visualize these things with. So a light microscope is going to be pretty good from about 1 to 2 millimeters all the way down to 200 nanometers maybe. At that point we lose resolution. We just can't see things as being separate. Everything gets fuzzy. We can't make them big enough, so we don't see anything at all basically. So we could see, you know, mitochondria down here, a couple of microns in diameter, a couple of microns long, with a very, very good light microscope and the right staining techniques. If we get smaller than that down in the realm of the viruses down here, there are some exceptions. There are some giant viruses that have been discovered. If we're down in the realm of viruses 100 nanometers down to maybe 5 nanometers in diameter, then we've got to use an electron microscope. So as what I want you to take away from here is that you should have a relative understanding of the sizes of things. You should probably know that a mitochondrion is about 1 to 2 micrometers in length, and then how many times bigger is a eukaryotic cell, you know, how big is that compared to a frog egg, and then going the other way, the rough size of a virus relative to ribosomes, that kind of thing. So it's a relative scale of things, but then learn a couple of benchmarks. A good one is the diameter, is the width of a membrane at 7 to 8 nanometers, size of some of the common organelles, size of E. coli at 1 to 2 micrometers in length, and then the rough sizes of cells. Good idea to have an idea of these things and be able to put them in relative order. So that gives us a little bit of an idea about scale. So what I want to do now is turn our attention back to microscopy, because microscopes are what we use to see these very, very small things. So this was, the microscope was the earliest tool of the cytologists. These are people who study tissues and cells. And so using the light microscope, we discovered the structures, the nuclei, the mitochondria, the chloroplasts, those kinds of structures were discovered basically using microscopes. The most basic type of microscopy you can do is called bright field microscopy, and this is where you just have a source of white light and you shine it, you illuminate a sample, and the light is transmitted through the sample, and there's some reflection and there's some refraction, and you end up with an image based on the transmission of the light, and the light that's not transmitted is reflected or absorbed or refracted elsewhere. And so in the top left here is what we're seeing is these are probably cheek epithelial cells or something like that, a little tiny nucleus in here, and you don't really see much. That's because there's no inherent contrast, there's no pigment or anything like that. Now if you want to stain structures, or if you want to see specific structures, then you've got to stain them or dye them, and that's what's been done here with this bright field microscopy on the right. So these have been stained with a stain that, I'll be honest, I don't know what it is, but it lets us start to see discrete structures. We can get a better idea of where the cell membrane is, and we can start to see internal structures. Now, because there's very little contrast in these images, we then have special types, or in these cells, we have special types of microscopy that help us magnify or amplify, I guess is a better word, any subtle contrast differences. So the first one of these is called phase contrast, and what phase contrast microscopy does is it uses these special optics which align different wavelengths so that you get peaks and troughs, and the end result is that you get much, much better contrast. So if you compare the bright field at the top with the phase contrast down here, same cell type, you can see that the phase contrast gives us much, much better resolution. So most cell biology labs will be using phase contrast microscopes if they're looking at unstained cells. A little bit of a twist on that is a technique called differential interference contrast microscopy, DIC microscopy, and again that just uses some fancy optics to increase contrast, and again you get a slightly different image comparing the DIC to the phase and comparing them both to bright field. So those are some basic types of microscopy. Getting a little bit more complicated, we can talk about fluorescence microscopy. Now this is where you stain specific cellular structures using fluorescent dyes and molecules. The other thing you can do is you can take an antibody which recognizes a particular cellular structure, and you can add a fluorescent dye to that antibody, and when you treat the cells with that antibody, it goes and it binds to the structure that it's specific for, and then you have a special microscope which illuminates the sample with a laser, and that excites the fluorophore, and then the fluorophore emits a wavelength of light which you can detect using a bunch of filters and fancy electronics and computing. A twist on that is confocal microscopy, and confocal microscopy works like fluorescence microscopy, where you take a sample and you stain it or you dye it with a fluorophore, something which fluoresces when you illuminate it with a laser. But what happens in a confocal microscope is you take lots and lots and lots and lots and lots of thin slices. These are optical slices, so you focus on one plane in the image, and then the microscope slightly refocuses and takes another image, and refocuses and takes another image, and you get a stack of images, and that can be over the course of maybe 1, 2, 5 microns, and then you can squish them all down, and then you get very, very good, very high resolution three-dimensional images. And that's called confocal microscopy, and that's very powerful, and you see that being used a lot. A lot of the images that I show you of cells that have been stained will actually be confocal images. And then of course there is the ever popular electron microscopy. Two types of this: transmission electron microscopy and scanning electron microscopy. Transmission is used for looking at internal structures, and scanning is good for looking at surface structures. So the development of the EM led to massive improvements in resolution because of the short wavelengths of the electron. So light was replaced by an electron beam, and the lenses were replaced by magnets, which were used to then focus the electron beams on the sample. So you can get resolutions of up to 2 nanometers. So you can see two things as being discrete if they're 2 nanometers apart. So if you've got 2 ribosomes that are 2 nanometers apart, you could tell them as being discrete. And if you look at this image on the right hand side here, this is a mitochondria in the middle, and then this is rough ER over here, and you can probably make out the little dots. Those are ribosomes. You can actually visualize these things with a good transmission electron micrograph. Magnifications, we're talking up to 100,000 times. So a good lab microscope in a general biology lab is 1,000 times magnification. 100,000 times bigger than the actual structure we're looking at. So here we've got a mitochondria that maybe that's 500 nanometers, so 500, 100, it's about 2 micrometers in length, 2.2 micrometers in length, and we can see the individual cristae. Those are the invaginations of the inner membrane in the mitochondria, remember. So we can see those individual invaginations of the cristae, which is phenomenal. Over on the left here, this is intestinal epithelia. So we're looking at microvilli here on the surface of the intestinal epithelia, so we can visualize those structures as well. Just think back to how small these things are that we can now look at using these types of microscopy. At the bottom here, we've got a breast cancer cell which has been scanned with an SEM, and then some pollen from the sunflower, and you can see you get these beautiful, fantastic three-dimensional images. So SEM is used for surfaces usually, and then TEM, transmission electron microscopy, very, very good for internal structures. So that's thinking a lot about cell sizes and using microscopes to visualize, to actually look at and describe structures. But a lot of cell biology is studied using other tools, and one of the main tools is biochemistry, and we learn a lot about how cells work from doing biochemistry now. So biochemistry is really kind of a separate field unto itself, but it's understanding some basics about biochemistry is important for understanding cell biology. So I always have the perspective about biology that it's really just applied chemistry. That's all it is. Cells follow the same physics and the same chemistry laws as do physicists and chemists. So it's good to think about biology as applied chemistry. Much of what we know about cells and how they work is learned from studying what's called cell-free extracts. So you can take a cell, you can homogenize it and break it open and take the components and study the biochemistry of those components. We can also then take parts of the cell, the mitochondria, the chloroplasts, and again study those in extracts to look to see how certain reactions proceed. So here's a nice example, and this example uses radioactive isotopes as well, and radioactive isotopes have been used for many, many, many years to trace certain molecules through biochemical processes in cells. So what we've got going on here in this experiment was, well, let me tell you what this experiment was. This experiment was performed many years ago to identify the main steps, and the sequencing of the steps in the Calvin cycle. So that's it's the dark cycle, or we should probably call it the Calvin cycle. And so what we've got going on here is this this balloon flask here is containing an algae called chlorella, and it's being illuminated by a lamp over here. So there's photosynthesis going on here in this algae, and what we're doing is we're pumping radioactive carbon dioxide into the flask, and so the carbon dioxide gets fixed using the Calvin cycle over here on the right. And as what we do is at time intervals after we start pumping in the radioactive carbon dioxide, we collect drops of the chlorella into alcohol, and when the chlorella hit the alcohol, they basically stop all of their biochemistry. And as what we can then do is take some of the alcohol which has got the chlorella in it, and we can break open those cells, isolate the internal cell components, all of the molecules which were being produced, and we can then say to ourselves, well, which molecule did the carbon dioxide go into first? When this experiment's actually done, you can separate the molecules using thin layer or paper chromatography. So we've got a little spot of our extracted pigment from our chlorella, and we can separate out the pigments, and so the pigments would separate out, obviously, and then all the other molecules also stood out. So if you remember from general biology, ribulose bisphosphate is a molecule in the Calvin cycle, 3-phosphoglycerate, glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate, all molecules involved in the Calvin cycle. So what's going to happen here is, if a molecule is very early in the Calvin cycle, then the carbon dioxide will go into that molecule first. So whichever one of these is radioactive at the beginning of the experiment, that tells us this is the first molecule that carbon dioxide gets added to, and then, as you know from the Calvin cycle, the carbon gets shuffled around until it's eventually released to glyceraldehyde 3-phosphate. And so you can basically take the chromatography paper, you turn it on its side, and you run it through another solvent, and so we've got this little kind of two-dimensional map of all these molecules and pigments and things, and you put it on a piece of X-ray film, and wherever you get a little black dot on the X-ray film, that's the first place the radioactive isotope went. And so by doing this over the course of a couple of hours, is what you can see is the carbon dioxide starts off in one molecule, then it moves to another molecule, then another molecule, and another molecule, and this basically orders the sequence of steps in the Calvin cycle. And so when you look at the Calvin cycle and you see this, this complicated cycle, and you often wonder yourself, how on earth was this figured out? It's using techniques like this. So we learn about the cell biology of photosynthesis using this in vitro biochemical technique, and so these are complicated things to work out, and we can't do everything in whole cells, so we have to take pieces of cells. So cell biology is often considered to be a reductionist field, because we break things down to the smallest components we can and then try and build the bigger picture back up again. That's kind of how cell biology is thought about in many ways. Another biochemical technique which you may not have encountered, which is important, is electrophoresis. And there are lots of different types of electrophoresis. This is SDS polyacrylamide gel electrophoresis, and this is used to separate proteins. So if we took a cell and we broke the cells open, so we take a bunch of cells and we smush them up basically and take the cell contents. If we wanted to look at all the different proteins in the cell, so we wanted to start doing some kind of proteomic analysis, one of the things we would do is separate those proteins based on their molecular mass. And so the proteins are heated up in a variety of different chemicals, and then they're applied to the top of this gel, and then we run the proteins through the gel based on a current we apply to the gel, and the small ones end up at the bottom and the big ones end up at the top, and then we can use a whole range of techniques to look to see whether there are different proteins present and which proteins are absent, do any change in abundance. So again, we've taken a whole cell and we've broken it apart, and we're looking at individual pieces, and we take the pieces and we try and build them up into more complicated pictures. So the only reason I'm introducing this to you is because this technique and other techniques that as we go through the course, they're going to become more and more important to understand how we know what we actually know about the behavior of cells. So let's wrap that up there. Next time we'll talk about immunofluorescence, which is a way of looking at certain structures inside cells using fluorescent molecules, and I'll see you next time.